Hi everyone, and welcome to the fourth TIA Centre seminar of the academic year. So for those of you online that are new to our seminars, we try and invite researchers and academics from across the globe to present new and exciting work. And today we're delighted to be joined online by Dr. Pingjun Chen. Pingjun is an instructor committed to computational pathology at the University of Texas MD Anderson Cancer Center, where he conducted postdoctoral training from 2020 to 2023, before he earned BS and MS degrees in software engineering and medical image analysis from the Dalian University of Technology in China in 2012 and 2015. He received a PhD degree in biomedical engineering from the University of Florida in 2020. His research concentrates on developing cutting-edge computational models and frameworks to advance cancer prevention, detection, diagnosis, and treatment. He has published over 30 peer-reviewed journal and conference articles in these research areas. Dr. Chen aims to harmonise the perspectives of pathologists, oncologists, biologists, and other computational scientists towards developing robust solutions to address critical unmet needs for making cancer history. The title of Pingjun's talk is Pathomic Features Reveal Immune and Molecular Evolution from Lung Preneoplasia to Invasive Adenocarcinoma. And Pingjun, yeah, thank you again for joining us. And when you're ready, please feel free to start presenting.
Hi, everyone. Thanks, Adam, for your kind introduction of me. Yeah, it's a great honor to me to have this chance to introduce my research here at the TIA Center seminar. And as Adam introduced, so my talk today is Pathomic Features Reveal Immune and Molecular Evolution from non-preneoplasia to invasive adenocarcinoma. This paper was recently published in Modern Pathology. So first, some background. Although in the United States, lung cancer is not the cancer type with the most new incidences, but lung cancer is the leading cause of cancer mortality in the United States. So from the 2023 data, as we can see on both males and females, lung and bronchus cancer is the top of the estimated deaths. Like even in females, it's like higher compared to the breast cancer. So in this study, we mainly focused on the non-adenocarcinoma, which is a subtype of non-small cell lung cancer. The lung adenocarcinoma accounts nearly like 30 to 40% of all cancer cases. So in this figure, shows the steps of the early adenocarcinoma carcinogenesis. So basically from so firstly, in the normal stage, so all the epithelial cells are functionally normally, and then it comes to the atypical adenomatous hyperplasia stage. In this stage, some epithelial cells becomes atypical, while in this stage is still not cancer stages, is not cancer yet, but from this stage, some of the epithelial cells will become cancerous. And like then comes to the adenocarcinoma in situ, and we call it AIS. So in this stage, it is called the beginning of cancer. And from AIS, some of the cancerous epithelial cells will gradually enter into the bloodstream or lymphocytic system. And from here, more and more cancerous cells will through the lymphatic system and bloodstream go to other organs, namely metastasis. So this would become the invasive adenocarcinoma. So here is like the shows the overall process of the carcinogenesis. So so why we are focusing on the pre-invasive cancer in this study? So it's no surprise that localized original lung cancers, so basically this pre-cancers have a far better survival outcome compared to this metastatic cancer types. And another data in the this is the data from actually from Taiwan. So in the past 20 years, since the widespread implementation of lung cancer screening, more and more localized staged lung cancer has been found. So under this situation, if we can potentially identify biomarkers that can intercept this early stages lung cancers, then we can push up the lung cancer survival curve significantly up. In that case, it would be very helpful for the lung cancer prevention. So understanding the lung adenocarcinoma early carcinogenesis gradually become a hot topic since roughly 5 years ago. So in this 2019 article published in the Journal of Thoracic Oncology, this group studies the genomic landscape and the immune microenvironment features of pre-invasive and early invasive lung adenocarcinoma. And this study from our group, led by Dr. Xin Hu, she studied the evolution of DNA methylome from precancerous lesions to invasive lung adenocarcinoma. And there are also other groups using single-cell RNA sequencing and spatial transcriptomics try to delineate the dynamic evolution from the preneoplasia to invasive lung adenocarcinoma. So as you can see, so most of the studies is trying to use genomic or RNA sequencing, like this transcriptomic, to study this early carcinogenesis phenomenon. And then our group thinking, can we try using computational pathology to study this problem? So then comes to the computational pathology. So in order to use computational pathology, the first step is using whole cell scanners to convert glass slides into digital slides. So with digital slides, firstly, it can enable computer-based management, inspection, sharing, teaching, etc. And most importantly, the digitized slides provide the opportunities to applying artificial intelligence to pathology study. So most available computational pathologies applications can be categorized into the following three groups. So the most large group is diagnostics. So basically tumor detection, subtyping or grading. And another group is prognostics, prediction the patient survival outcome, and also possibly the cancer relapse, and also maybe toxicity of certain treatment. And also for the therapeutics, for the treatment selection and optimization. And in this study, actually, we able to using computational pathology to decipher tumor microenvironment. To the best of our knowledge, no prior studies had yet investigated the cellular level lung precancerous progression using computational pathology. So thus, in this study, we developed a computational pipeline to study the tumor and the immune progression from preneoplasia to invasive adenocarcinoma. So here, a very simple, first a very simple introduction of this pipeline. So first, to curate the slides and ROIs, and then we conduct cell segmentation and recognition. And after the cell recognition, then we try to extract pathomic features from each ROI, and we extract several groups of different features, and then we study this pathomic feature progression trends along with the cancer evolution process. And at the same time, we also conduct the correlation studies between some molecular biomarkers with these pathomic features. in order to implement this developed pipeline, and this shows the created patient's characteristics. We create 3 study cohorts. Actually, the patients from Japan, China, and the United States, and this is like their roughly like their characteristics on sex, age, and tobacco usage because of the lung cancer. So tobacco usage is a very important factor. So as you can see, the number of patients is like quite small here, because this is like to study the pre-cancer. It's really very challenging to get to get the tissue samples. And first, I want to introduce the slides acquisition and annotation process. So for all the slides are shipped to our institute, namely MD Anderson Cancer Center. So then we digitized with the same scanner, so namely Apero 82 at the same magnification 20x. And first we ask one pathologist to conduct a quality check to exclude slides with very evident, very like severe evident artifacts. And then we ask 2 pathologists to confirm the diagnosis of each slide. And here are several things to note. So for one patient, it can have multiple different slides, and for different slides, for the same patient, different slides can even have different diagnosis. And also on the same slide, it can contain multiple different morphological patterns. For example, like we mentioned, like from normal to AIS to AH to AIS to finally to ADC. ADC slide can contain the morphology patterns of the previous stages. For example, ADC can contain AH or AIS patterns. So with this consideration, we adopt a ROI-based manner to analyze tumor and immune progression to study the lung neoplasia. So so first we request one pathologist to annotate diagnostically informative ROIs. Then these ROIs are checked by 3 other pathologists to give their diagnosis separately. And we're only using those ROIs with consistently diagnosed pathological stages. And another thing is that we aim to limit the ROI size between the size, both height and width, between 500 pixels and 5,000 pixels. So like we use the smallest ones to ensure the ROI contains sufficient number of cells. And for the maximum is like we try to limit the size of the ROI to mitigate the computational burden. And this slide shows, this table shows all this data we obtained. So although even we have 3 cohort data, but the patient number is actually less than 100. But we annotated more than 600 ROIs from 162 slides. And one thing to note here, actually for the China cohort, we don't have normal slides, but we can annotate normal ROIs from more severe stages, namely, for example, AH, AIS, like for example. So that's the case why, like for this Japan and China, even though there's no normal slides, but we still can create a quite large number of ROIs. So this is like all this. So what this 669 ROI data are the ones we use for the following analysis.
Sorry, Pingjun, can I ask a quick question?
Yeah, sure.
So for instance, in the first row, so where there's 9 slides from the USA, but no, for normal that is, but no patient, is that the case that the tissue is from a cancerous tissue, but then this one slide from that was normal?
Yes, so it's like for so basically, like for example, this normal ROIs is from like the raised slides. For example, the slides can be AH, but it still contain normal ROIs.
Okay,
So that's like how we can obtain the normal ROIs from here.
brilliant. Thank you.
And since we will conduct a cellular level analysis, and then the first step is cell segmentation and classification. So as we use H&E staining, so the cell membrane is unclear. So basically, it's like the cell we mentioned here actually is used interchangeable with nuclear. And after like we did quite a lot of literature review, and also we did quite a lot of experiments, and we found the HOVNet developed by the TIA center. So we found this model shows the best performance on the nuclear segmentation performance, and this I show one example. So our in-house pathologists randomly check dozens of ROIs, and they find like the nuclear segmentation quality is amazing based on their saying. And also we have done a small scale quantitative evaluation, and we through our experiment we found the DICE score of the nuclear segmentation is like over 0.8, so which is is super good. So we assume under this situation the nuclear segmentation would be good enough for the downstream analysis. And the pre-trained model we used is pre-trained on the PanNuc dataset. So this dataset covering 19 different TCGA tissue types. Of course, lung cancer is one type of it, and also HOVNet contains the classification branch, and it can classify each nuclear into the following 6 categories. While like all pathologists like check their nuclear classification results, they think the classification results is cannot satisfy their requirement. So, and also based on the discussion of our in-house pathologists for this lung cancer problem, they propose this hierarchical cell type categories. So for the segmented cells, so they think it's more reasonable to first split them into lymphocytes and atypical epithelial cells, and like we use these other cells to cover many other cell types. So the reason we use here, like we're using, instead of using cancer cells, and here and we use atypical epithelial cells, is like we want to consider the AAH stage. So since like AAH stage is the early stage, it should be not contain any tumor cells. So if like we use the tumor cells here, it will be problematic. So that's the reason we use atypical epithelials here to cover the tumor cells and some atypical pneumocytes. And then like we aim to classify all the segmented cells into these 3 different categories, namely AEC, lymphocytes, and other cells. So so based on the domain knowledge from pathologists, they usually recognize the lymphocytes based on 3 morphology features. So they say small, dark, and round. So then we like have a very simple idea to extract features based on these 3 morphology characteristics. Namely, we propose using area, intensity, and roundness for the cell classification. And since we use the intensity features here, and one thing we need to consider is the stain consistency. Since we get the slides from 3 different countries, so like here we compare like 3 raw ADC ROIs. If you have a look, you can see there's quite obvious color inconsistency between them. And in order to reduce this problem, we adopt the very commonly used Marchenko stain normalization algorithm to do the stain normalization. So after this normalization problem, we think this stain inconsistency is relieved. But I shall say that definitely there will still stain inconsistencies inside of this ROIs. And to build a cell classifier, so we asked our in-house pathologists to annotate small number of cells. So actually we choose more than 10 ROIs from both Japan and US cohort, and like for 3 different cell types, this is the number of the cells they help annotated. It's quite a small number of cells, so so its annotation is usually not opinion work. They are usually just willing to annotate a small number of cells. And before we training the cell classifiers, so what we first do is compare the feature distribution. So here we compare the area, intensity, and roundness. So if we compare the area from these 3 different cell types from these 2 cohorts, they are quite consistent. But when it comes to the intensity, as we can see, for this cells from 2 different cohorts, you can see there's more discrepancies between them in all 3 different cell types. And on the roundness, and the AEC and the lymphocytes are quite consistent, but for the other cells, it's more big difference. One reason for this other cells is more prominent is, I guess, is like because other cells contain a far more diverse types of cells, which could lead to this issue. And with this cell annotations, we using XGBoost to train the cell classifier, and we tried evaluate in using 5 different ways. So the first way is like we only using the Japan cells to conduct the 5-fold cross validation. Similarly for the USA data, and also we combine them to do the 5-fold cross validation. The following two the following two is like we try the cross evaluation. So namely, the model trained using Japan cells to evaluate on the USA cells, and also using the model trained using USA data to evaluate on the Japan cells. So as we can say, if building the cells from 1 cohort, and the accuracy is quite high, like nearly 0.9, and also like with combining is also quite high. But when it's evaluated on the cells from other cohort, the performance would decrease. Even though like the Japan model on USA cells don't decrease that much, but it decrease, and the USA data trained model decreased quite more obviously on the Japan cells. So considering this scenario, we believe using integrating all the cells together to train a model would give more robust performance, since it contains more diverse cell. It contains cells from more diverse sources, and also like if like on this fusion dataset, the standard deviation is lower compared to both A and B. So that's the reason we use this fused data to train the XGBoost classifier for the following analysis. So with this trained cell classifier, we apply to all the segmented cells on all the annotated ROIs. So after that, for each ROI, we have all the cells segmented and annotated into these 3 different tabs. And for the cell recognition, we do another round of evaluation. So we ask our pathologist to conduct a rough estimation on the precision score. So basically, for each cell type, with this model, we know like all these cells predict to a certain type, and inside those predict certain types, we will check what are the true positive ones. So namely, is the precision. The reason we choose this way is easy for pathologists to conduct this estimation, and we ask the pathologist to roughly like split them into 4 categories. So if they are like in the man, like roughly the precision like larger than 0.9 is like, so mostly are good, then it's like near perfect. If like roughly most of, if like you can obviously say some wrong, then we say it's like quite good. But like say like a large number is like maybe more numbers are wrong, then it's acceptable. But if like nearly half is wrong, then we think it's poor. And we ask 2 pathologists to do this evaluation. And so for these numbers, after this cohort name is like the number of ROIs to do the evaluation. So for the Japan, we ask a pathologist to evaluate on 44 ROIs. So from their evaluation, so you can say like for the atypical epithelial cells and lymphocytes, most of them, they think the precision are quite good. And this is like on all these different data sets on both Japan, China, and USA. And for the another pathologist, similar things. But on the other cells, they think the precision score is not that good. So like this is also quite consistent. So under this situation, so we would think like using our classifier, the typical epithelial cells and lymphocytes are more robustly recognized, but for the other cell types, the performance is not acceptable. And then we would mainly using these 2 cell types for the following feature extraction. And then the next step is pathomic feature extraction part. So here we extract 4 different groups of features. So the first 2 groups are very easy to understand. So first, the cell ratio, and as I mentioned, because the AEC and the lymphocytes are the recognition performance are more strong, and we only consider them. So for the cell ratio, we only consider this 2, and for the cell density is like it measures the compactness of the cells inside each ROI. And for the cell spatial entropy and the embedded map texture, I will explain in the following 2 slides.
So can you ask a question? Sorry.
Sure.
So what's the difference between the cell ratio and the cell density here? Sorry.
Oh, so the cell ratio is like divided by the, like for example, the number of certain type of cells divided by the total all the cell numbers is a ratio. I guess it's like, sorry, I think proportion will be, sorry, I forget to check. Proportion will be more reasonable. And for the density, is the number of cells divided by the total area. So that means it is like the compactness of the cells in each ROI. So basically, this is like the relative abundance, and this is like the compactness in each ROI.
Thank you.
And here, like for the cell spatial entropy. So I guess like I think most of the audience should be familiar with Shannon entropy. So the Shannon entropy basically consider the ratio of several entities. I guess like this is example like showing like for these 3 different categories, because their ratios of these 3 different types are the same. So that's the reason the Shannon entropy is the same value. for the spatial entropy, we want to also to consider their position information. So basically, for each point here, what is the like XY location inside of this, inside of, for example, in our case, inside the ROI. So that's the reason like the spatial entropy would be different since the location of the different points are different. So inside this spatial entropy package, they provided two ways to calculate the spatial entropy. So in this study, we adopt the second one, the R-Terrace entropy. So as you can compare, if you say like this, different cell types are well separated in this case. So this R-Terrace entropy is like relatively smaller compared to this one when the cells are more mixed. So that's like a way. Here I give the example, like if like the cells like they are more separated, then the spatial entropy is slower. While when the cell types are more mixed together, then the spatial entropy is higher. So here we want to consider both the cell numbers and also the cell locations, and we aim to use this kind of like to measure the intratumor heterogeneity of each ROI. And as for the embedded map texture features, so here also we already consider the atypical epithelial cells and the lymphocytes. So for this, how we do it is we first setting a sliding window. For example, the size is, for example, 100 by 100, and then we consider for each window how many epithelial cells, typical epithelial cells inside this window, and how many lymphocytes inside this window, and we encode this value into this point as a number. So we do this by sliding windows. In this case, this embedded map would be, if like this is like 100 by 100, then this embedded map would be 100 less in both height and width. So it becomes a far smaller map. Because like for this map, it will be a green scale image, and then we first calculate the green level occurrence matrix, namely the GLCM, and then from the GLCM we extract the texture features. Here, like actually, the GLCM has many different features. Here we're mainly using contrast and energy. So we through this way to try to compress a large ROI, a relatively large ROI, to a far more smaller ones, and then extract the texture features. And also, like in this way, we can kind of like keep some of the local information. Still, some of the spatial information is kept in this texture features. And overall, we extracted non-pathomic features. We aim to use these features to study the evolution trends. So we aim to use these features to decode the non-carcinogenesis. So we aim to evaluate their features, mean value, and their overall distribution within each diagnosis stage to assess the progression trends from normal to ADC. And we also try to compare different subgroups. And last, we will do some correlations with molecular biomarkers. So first, let's have a rough look of the AEC proportion density trends. So first, let's have a rough look of here. So it's very obvious to see like the proportion of this epithelial cells is increased, and also the density increase from normal to ADC. But we can also find for each stage, there's quite large variations, and between two neighboring stages, there are quite high overlap between them. So we would think this is because of the heterogeneity between different pathological stages. And also, if we compare like different race. So here, like for the US data, actually we filtered them through the race. We only keep those with race white. Of course, USA has other races, but we only keep those patients with race white. And for both Japan and China cohort, we label them as Asia cohort. And like for this, when we compare their this AEC proportion, so for the normal and MI and ADC seems like they are more consistent, but like for this AAH and AIS, and this white population shows higher values. And on the AEC density, the late 4 stages shows on both white population shows higher values. And also like if we compare like the current and former smoker versus never smoker, so the smoker group shows a higher atypical epithelial cells proportion. But like on this density is the late 3 stages. And here we also compare like the lymphocytes proportion and density trends. So if like say for the AEC proportion, you say both the proportion and density increase. But for the lymphocytes, the proportion gradually decrease, but the density increase. So that's the reason it's quite from this lymphocytes comparison of the proportion and density is quite important to consider both features. They are not necessarily highly correlated. And also like if like in this lymphocytes, the late 3 stages, their values are quite in a very narrow range. They are quite consistent. And also like if we compare like this white and Asia cohorts, we see this Asia cohorts seem to have a higher lymphocytes proportion. And also like here on the lymphocytes density, the white cohort shows a higher density. But when we compare like the current and former smoker versus never smoker, we see most of them are no difference. So that shows there might be something like on the lymphocytes happens, but I'm not sure that there might some difference. And also here we are using the arterial entropy to evaluate the trends. So as I mentioned before, because this arterial entropy, we consider both the cell abundance and the location. We aim to use it as like a measurement of intratumor heterogeneity. So from this figure is like we can see like from normal to ADC, this arterial entropy gradually increases. So we would roughly interpret the intratumor heterogeneity increases. But of course, there are quite high overlap even between the normal and ADC. And here we can kind of like using like the one like the physical law is like entropy always increases, is kind of like from the normal to the invasive adenocarcinoma is this is also heard from this in this neoplasia progression process. And another thing here I want to mention, like when we compare like the current and former smoker versus never smoker, it's like the smoker cohort shows higher intratumor, shows higher arterial entropy compared with never smokers. So that might means smoking can lead to worse outcome. And that's like one of my hypotheses. And here like is the embedded map texture features trends. So the GLCM contrast mainly represents the local green level variations. So basically a higher contrast value indicates the presence of either edges or wrinkled structures inside of the GLCM matrix. And the energy measures the uniformity of green level distributions. So if a lower energy corresponds corresponds to a large number of green levels, namely lower uniformity. So from the biology perspective, so as ROS progress from normal to advanced stages, so the number of atypical epithelial cells will increase, and then more atypical epithelial cells will be scattered over the ROS, and it will be forming more edges and wrinkle structures, and thus the contrast likely to increase. But as the stages increase, the embedded map would be show more diverse distributions among local regions and causing low uniformity, thus causing lower energy values. So that's like the reason, like we try to interpret that why the AEC contrast increase steadily while the AEC energy decrease steadily from normal to ADC. And as for the lymphocytes, the trend is a little bit interesting. The first will be increase and then will be decrease. And while the lymphocyte energy will be at the reverse, we don't know how to interpret this yet. So this is roughly some of the feature trends analysis. And we also do a pathomic feature correlation study with some molecular biomarkers. So we select a subset of the cohorts matched slides to assessing their molecular markers using her exome sequencing, and we measured 3 biomarkers: so the tumor mutation burden, allelic imbalance frequency, and copy number variation. So I'm not biology background, so actually I don't like the details of this list, but basically it like means if like it's more severe, if the cancer is more severe, so this number would be higher. So when we conduct the correlation, we use the Spearman's rank correlation between these 9 pathomic features with these 3 different molecular biomarkers. So it's like and also like here we set a threshold 0.3. If the value is less, if the absolute value of the correlation value is less than 0.3, we set them as 0. That's so that to make other high correlation values to be more prominent. So as we can see here, like the atypical epithelial cells, namely we can roughly understand this is tumor cells, its proportion shows positive correlations with this 3, like both this tumor mutation burden and this AI burden on the CNV burden. And similarly for this arterial entropy, so basically I try to interpret it as a intratumor heterogeneity. It also shows quite strong correlation with this tumor mutation burden and this allelic imbalance frequency. So from here, like we are thinking like this pathomic features from H&E staining shows some strong correlation with these molecular markers. Some limitations of this study. So firstly, the cohort used in this study is quite small, even though we annotated like more than 600 ROS, but the number of patients, even from 3 different countries, but the patient cohort is not big. If like a large patient cohort would make this discoveries to be more interesting. And also like for the cell classification, so both granularity and accuracy may need further improvements and validations. And also regarding the cell classification, so our in-house pathologists, they assume that we need to consider the context information around the nucleus instead of just using the nucleus, because when they evaluate the cell type, definitely they will consider their context. That's one thing to consider when build the cell classifier. And also, like as I mentioned, there could be some tissue inconsistency of all the slides, even though we conducted stain normalization. So with tissue preparation standardization, I guess it will also help alleviate these issues. And some takeaways. So this study, I guess, demonstrate some feasibility and potential of using low-cost routine H&E staining to study tumor microenvironment. I think this pipeline can apply to other cancer types, but like the major module need to change is the cell classification part. And also for this study, it shows the foundation rules of the six cell segmentation and the recognition. I think that's the credit goes to the developing of deep learning and machine learning models we need like this strong cell segmentation and recognition in order to conduct this analysis. Another thing is regarding the model generalization and specialization. So for the nuclear segmentation, I guess HOVRNet demonstrates super strong generalization capability. But when it comes to specific tumor types, for example, like especially goes to the cell classification, we might need specialization models instead of maybe, I guess, a general models might not work well. So that is like specialization is also quite important. That requires the involvement of domain expert for this help. And another part is in order for the biology understanding, handcrafted and features with interpretability is quite important, since pathologists, they always want to understand what all these features mean, even though like for me it's still quite challenging for me to explain those textual features from those embedded maps. And for this study, we made the code and data public available on this two websites. So basically, all these results I showed in this paper should be able to reproduce from the code and the data. So feel free to try if you have time. And so I would acknowledge the multidisciplinary team and the collaborators in conducting this research. So yeah, including like pathologists, oncologists, and biologists, and of course we need computer scientists. So those names with bolded is the senior authors in the study, and those names starting with star is the co-first authors of this study. So this is the end of this presentation, and I'd like to take any questions, if any. Yeah.
Brilliant. Well, thank you very much, Pingjun, for a really clear and interesting talk. So yeah, with that, we'll open up the floor to questions. So does anyone online or in the lab have any questions to start us off with?
Yeah, can I ask a question?
Sorry, Pingjun, can you hear that okay? Or
Yes, I can hear.
Brilliant.
So thank you very much. Really interesting talk. And you might have answered this, but based on the plots that you presented for density of lymphocytes throughout the cancer stages, so you suggest that one of the main reasons for cancer progression is lack of lymphocytes. Is that right?
To be honest, so I don't have sufficient knowledge to answer this question. But like actually, one thing I can share, like actually I also discussed this point with my collaborators, biologists. So basically, like one idea he shared with me, one very important factor is based on where you annotate the ROI. So if you annotate the kind of like the how to say, if you annotate the tumor core, then more likely the lymphocytes the number of lymphocytes in that area would be far less. But if you annotate the kind of like the boundary of the you annotate the ROI from the tumor boundaries on that area, then the lymphocytes, both the proportion and the density could be much higher. So that's like one thing we are always thinking, like for the understand for this kind of measurement can be quite biased because it's severely affected by which area you annotated the ROI.
See.
Brilliant, thank you. So the next question, I suppose, for me is that, so obviously you've got these 9 features that seem to be quite good at discriminating between the different stages. Have you actually tried to build a model to use these features to perform the individual classifications?
Actually, we do try, but actually when we since like in this study we have 5 stages. When we trying to classify like all this into 5 separate stages, of course maybe this task is more challenging. So, but the performance actually is not very is not very promising. It's like kind of like, as far as I can remember, it's like for 5 categories, it's less than 60%. And then we consider, okay, maybe we can merge like the middle 3, and then we make it into a 3-way classification problem, basically normal, and then this pre-cancer, and then ADC. And this scenario, the performance is kind of like more than 80%, but still we think it's like it's not a very convincing. So that's the reason we don't report this classification results. We do have some of this classification study. The one reason is like, as I explained, like is like there are quite strong overlap between them. So that makes it quite challenging to well separate them, even though we claim like we have nine features, yeah, but they all seem to have like quite a strong overlap.
Brilliant, thank you. And
yeah, and also also that maybe like one thing like speaks to the like heterogeneity of biology. So it's like and one of our collaborated like oncologists is usually suggest us like this is like we can think of it as a continuous process from normal to the ultimate to the ADC. Actually, it's hard to find the cutting point. So always like 2 neighboring stages, they will have some overlap. So it's so that is the case. So it's very hard to really, even for pathologists, sometimes they cannot find a consensus to say like which stage this ROI is. Yeah, but like for your question, like we try to do the classification, and the performance is like it's not as decent as we expected. Yeah.
Okay, and I suppose another question is in terms of the next stage maybe of this analysis, is it to try and find these ROIs automatically, or is it because obviously they're carefully selected by the pathologists at the moment, aren't they?
Yeah, I think actually this is a very interesting idea. So so after this study, we don't continue the study because we think like it's good to have a large cohort, but it's really hard to create more data sets. So that's the reason like we stopped the research here. But I guess like your idea is very interesting. If like we can automatically detect those ROIs, I guess that's a really a very interesting direction to go. Yeah, thanks for this suggestion.
No worries. And do we have any other questions from anyone? Oh, go ahead.
Yeah, I had 2 questions actually. One is like, have you done anything for the outlier values for the features? Because we don't see the flyers here. I was just wondering that.
For the outliers, okay, yeah, I guess they could have some, because if you see like there's like kind of like that's quite long, I guess, because I guess this is like this way of drawing this way, so it don't like showing that outliers. But if using another, I forget what that box plot, I guess because this volume plot, so maybe outliers don't show up. But if using box plot, I guess outliers can show up. I think definitely there will be outliers, especially consider it's such a long range.
Yeah. And my second question was, have you tried to perform any statistical tests to see if the distributions are like significantly different than each other?
Okay. Yeah, thanks for this question. So, this, actually, like, my mentor also asked me to conduct that. The reason I don't do this is because there are too many pairs. I think it's quite challenging for me to compare like all the pairs. And another reason I didn't do it is I mainly want to roughly check their trends. So like regarding the p-values, I guess maybe for this study, like from my understanding, maybe I don't think it's that important. So usually, like my understanding, like if we have if like the number of our ROIs is big, so it's easy to have the p-values. But I do get you. So usually, like p-value is a nice number to have. But I guess because I mainly want to roughly check the trend, so I don't calculate the p-value. Yeah.
Yeah. Thank you.
Brilliant. Well, thank you. So if we, I suppose, has anyone else got any questions, or shall I finish the final question? Brilliant. So one of the great things, obviously, about this study is that you've got 3 different data sets from 3 different countries across the world. So you obviously you said that you looked at stain normalization, but did you think about any other form of domain, any tools to sort of handle domain shift in general?
So of course, like there are multiple ways to do the stain normalization. The reason we choose this one is like it's like the most straightforward one, and also like based on this results looks like satisfying to me. So that's the reason we don't try other ones. Of course, I know I'm aware of there are many other ones, but it's hard to like say which one is better, because this is like a more subjective evaluation. So that's the reason like we adopted this the most like I shall say simple one, mechanical stain normalization. And one thing I want to share my experience here, like we also first try to using the HoverNet on the normalized ROI, but when we apply that, the nuclear segmentation performance is become far worse. The HoverNet segmentation on the raw slides, on the raw ROIs have very good performance, but on the stain normalized ROIs, the nuclear segmentation becomes far worse. So my understanding is like after this mechanical stain normalization, the contrast between the nuclear and non-nuclear areas, the contrast become less. So that's like one reason why the HoverNet nuclear segmentation becomes not as good as on the raw ROIs. If you guys interested, maybe you guys can have a try.
That's interesting to hear, because I mean, well, generally my experience has been that classification seems to improve if you do some form of normalization with HoverNet, but the segmentation is normally pretty reliable. Has anyone else had that sort of problem before in terms of worse segmentation results? But no, that's an interesting finding. So
yeah, if you had time, please give it a try. Yeah.
No, well, thank you very much, Pingjun. That's been a really interesting talk, and thank you very much for joining us at such an early time over there. So, and yeah, thank you everyone online for joining. So I'd just like to remind everyone that our next talk will be on the Monday, the 4th of December, where we'll be joined in person by Professor Jan Brosens from Warwick Med School. So I hope to see you all there. It's also a slightly different time. It's at 10:30 as opposed to 2, so it's a bit earlier. But no, thank you very much again, Pingjun, and I hope you have a good rest of the day. And thanks everyone online.
Thanks for having me. Bye.
See ya.
Thank you. Bye.